Hey everyone, so my senior honors thesis is about developing a fluorescent immunohistochemistry protocol for the muscle biology lab to use, and that'll allow us to study human skeletal muscle further. Before we delve into the specifics behind fluorescent immunohistochemistry, we first have to introduce muscles themselves. So humans have several different types of muscle in their body. There's cardiac muscle, which is in the heart, smooth muscle, which is in your vital organs like your stomach, and skeletal muscle. And skeletal muscles are the ones that you see when you flex your arm, for example. However, if you zoom into the muscle itself, then you see that it's broken into more and more tiny parts that contribute to its overall function. And what's so cool about these skeletal muscles is that they can get shorter and longer based on what you tell them to do. And my specific scope in this situation is to study the single fiber level of skeletal muscle tissue. And how is skeletal muscle function determined? So it's studied through two main parameters, its size and its contractile properties. On one hand, the size of the muscle fiber can be brought down to how thick it is. Like the thicker the fiber, the more force it's able to produce. Meanwhile, on the other hand, the MHC isoform found in the fiber, which is the myosin heavy chain isoform found in the fiber, determines how fast it contracts and how much force it produces. These MHC isoforms are basically protein variants found in the muscle fiber that dictate its properties. There are three main types of MHC isoforms found in human skeletal muscle tissue. There's MHC1, 2A, and 2X. And the main differences between these three MHC isoforms are its contractile velocity, which is how fast it contracts, and its force production. As you go from myosin heavy chain 1 to 2A to 2X, it contracts faster and with more force. In addition to these three main isoforms, we also see hybrids in human muscle tissue, like 1,2A fibers and 2A, 2X fibers, where we see multiple protein variants or isoforms on the same muscle fiber. So how do we determine the size and contractile properties? Here's where I come in. Fluorescent immunohistochemistry. So it's a method used to identify the presence of certain proteins on cells. It does this by first binding a primary antibody to the protein, and then a fluorescent secondary antibody to the primary antibody. So based on the primary and secondary antibodies that we use, we can fixate on different proteins on the cells, making colorful images like this one. Here, three proteins are highlighted and they're shown in green, red, and blue. From this, we see that the green is myosin heavy chain 1, which contracts the slowest and the least forcefully, then 2A, which is red, which contracts faster and more forcefully, and myosin heavy chain 2X, which contracts even faster and more forcefully. So why does this help? Like, why am I doing this? So based on the colors that we see and the analysis of the cross-sectional area, we can now better study the effects of aging, exercise, and disease on skeletal muscle function in the muscle biology lab. How does this protocol work? There are four main steps to the protocol, the first step being tissue preservation. So first, a muscle biopsy is taken from the thigh of participants, specifically the vastus lateralis, and put in OCT, which is a glue-like substance that helps preserve and stabilize the muscle section. It's then flash frozen in liquid nitrogen and put in the freezer at -80. Next, it's stuck in a glorified deli meat slicer, which is our like a cryostat, to make 6 micrometer cross sections of the muscle sample. Then these sections are stuck onto slides. And after sectioning, I come back into the lab and let it dry for 4 hours before doing my first incubation, my primary incubation, where I put the primary antibodies on the slide, stick it in a dehumidified cigar box, and leave it overnight. These primary antibodies are going to bind to the specific MHC isoforms found on the muscle fiber itself. Then I come back the following morning, wash off the primary antibodies, and incubate the sections with the secondary antibodies for an hour. These secondary antibodies will bind to the primary antibodies that were already there. I then put some mounting media on the slides, which preserves fluorescence, and put a cover slip on the sections to preserve them. Now that my slides are ready, I'm ready to image it. The fluorescent microscope we use is the Kion's BZX810, and I put my slides in the microscope, and through the software, I'm able to obtain 3 different images, each taken at a certain wavelength of light. And each wavelength corresponds to each MHC isoform, with the green being MHC1, which contracts the slowest and the least forcefully, 2A, which is red, and 2X, which is blue. Although this process may seem pretty straightforward, where we just bind the primary antibodies, then the secondary antibodies resulting in fluorescence, there have been one too many hiccups in the development of this protocol. This protocol was initially an adaptation of the University of Vermont protocol, but after we realized that it wasn't fully functional on our end, and we were only observing green on the fluorescent microscope, we went down several rabbit holes attempting to gain the optimal image. And upon starting, all we knew was that we were expected to see green, red, and blue on the images, and that's all we were pretty sure about. So here's our first issue, the lack of specific red binding. We always saw a generalized red stain but upon raising the exposure in the past, and after using more recent samples and buying new secondary antibodies, then we realized, wait, this is going away now, and we're actually seeing specific binding. So that's one issue that's one issue fixed. Another issue was we weren't seeing any blue binding. We initially seemed that we never seen any blue and that it looked like a lost cause. But then we realized that the images obtained from our fluorescent microscope at UMass are artificially colored and can be changed manually. This extended to us realizing that we've been matching each color to the wrong MHC isoform. So in a nutshell, this whole time we've been seeing the MHC2X fibers as red when we were told that it was going to be blue. And the green fibers that we've always seen in the past were actually our 2A fibers. And after we realized this, we adjusted the settings to ensure that our 2A fibers showed up as red and our 2X as blue. But what happened to our type 1 fibers? And now that we know that our issue was with our MHC1 fibers, we learned that we've been looking at it with the wrong microscope settings. And now we decided to buy some much more concentrated MHC1 primary antibodies and try that out with the correct microscope. Guess what? It worked after a year of trying and failing. So that's my thesis in a nutshell. I'd like to thank Professors Mark and Becky Miller for supporting and guiding me through this process, in addition to Professor James Chambers for assisting me with our microscope issues. I also thank the UMass Amherst Commonwealth Honors College for giving me the opportunity to conduct this research, and I extend a massive thank you to the members of the Muscle Biology Lab for the continued support over this past